Assalamu alaikum. I am Yumna Nadeem from section B of first semester. As per our designated assignment, I'll today be talking about experiment number 13, that is differentiate between short chain and long chain fatty acid containing fat sample. Starting off with what are fatty acids. A fatty acid is a carboxylic acid containing long aliphatic chain. It exists in the body in free state and can be either saturated or unsaturated. They can be classified into many types according to their length, saturation, branching, and number of carbons present. Based on length, they can be classified into short chain fatty acids, medium chain fatty acids, and long chain fatty acids. Our topics of interest here are short chain and long chain fatty acids. Short chain fatty acids contain 6 or lesser than 6 carbon atoms within their chain. Examples include acetate, that is a 2 carbon compound, propionate, that is a 3 carbon compound, and butyrate, that is a 4 carbon compound. Long chain fatty acids contain 12 or more than 12 carbon atoms. They can further be categorized into two main families, that is omega 6 fatty acids and omega 3 fatty acids. Examples include palmitic acid, that is a 16 carbon compound, and erucic acid, that is a 22 carbon compound, which can also be termed as a very long chain fatty acid. Moving on, the method devised to differentiate between short chain and long chain fatty acids within a sample requires calculation of saponification number. Saponification number or saponification value represents the number of milligrams of potassium hydroxide required to saponify 1 gram of fat which is hydrolyzed into free fatty acids, or the measure of content of ester linkages because it tells the amount of ester linkages present in the sample. It is also named as Kosthofer number after the German chemist of the 19th century, Kosthofer. The higher the saponification value, the lower the fatty acid's average length, the lower the molecular weight of the fat specified. Thus, we can say that saponification number also tells about the average length of the fatty acid and molecular weight of the fat specified. Each fat or oil has its own specific saponification number, thus, any deviation from that specific number indicates that impurity is present within the sample. Fats and oils with high saponification value are mostly preferred for soap making. Saponification number can be determined by the formula given. Here, B stands for volume of HCl used in Planck sample. S represents volume of HCl used in test sample. M is the molarity of HCl. 56.1 is the molecular weight of KOH, that is potassium hydroxide. And W here is the molecular weight of sample in grams. Proceeding further, here are some properties related to short chain fatty acids. They are produced in the human body by beneficial bacteria or friendly bacteria in human gut, mostly by fermentation of fiber in the colon, but also they can also be produced from all carbohydrates. High fiber containing foods like fruits, vegetables, legumes, and whole grains encourage their production. They might produce only 10% of the daily calorie need. Despite this, they are the main source of energy for colonocytes, that are cells lining the colon. About 95% of the short chain fatty acids in our body are acetate, propionate, and butyrate, with acetate being the most abundant SCF in our gut. Propionate is mainly involved in producing glucose in the liver. Acetate and butyrate are incorporated into other fatty acids and cholesterol. Properties of long chain fatty acids include that they dissolve readily in highly alkaline solutions, forming soaps. Palmitic and stearic acids are present in bulk within the animal tissues and most animal diets. Sources can be aquatic or plant sources, whereas LCFAs are abundantly also found in cyanobacteria and other microorganisms. Due to the large size, LCFAs bind up to proteins for covering long distances within the body or crossing cell membranes. This is termed as protein facilitated transport. They are a major component of most membrane phospholipids and play a significant role in membrane structure and function. LCFAs inhibit synthesis of vasoaggressive low density lipoproteins, more commonly known as LDLs. Docosahexaenoic acid, or DHA, is an omega 3 fatty acid that directly influences neurotransmitter biosynthesis, signal transduction, uptake of serotonin, binding of beta adrenergic and serotonergic receptors, and monoamine oxidase activity. That's it for my part of the presentation. Thank you.